Griffith's experiment. It helps to prove that DNA is the genetic material. Frederick Griffith actually selected a bacteria called as Streptococcus pneumoniae for his experiment to prove that DNA is the genetic material. Frederick Griffith selected Streptococcus pneumoniae, a bacteria which has two strains, R strain and S strain. So strains are a type of variety of bacteria. Here S strain denotes that it is smooth, R strain rough strain. So S strain, it is the R and the S strain are differentiated on the basis of a mucus layer present outer the cell wall. So on the outer part of the cell wall, if in the case of S strain, there is a mucus layer which is somewhat a slimy layer, and that layer appears somewhat smooth in appearance, means it is smooth in nature, hence it is called as smooth strain due to the presence of the mucus coat which is slimy in nature. But in case of R strain, there is no such kind of mucus coat, there is no such kind of mucus layer or slimy layer. So the cell wall itself is the outermost part of the cell, and that cell wall is rough in appearance, and that is why it is called as the rough strain, R strain. So the cell wall appears rough, R strain. In case of smooth, there is cell wall, but outer to the cell wall there is this slimy layer which is mucus in nature, and hence it is smooth appearing, and so it is called as S strain, smooth strain. So Griffith selected S and R strains for his experiment. Now what he did with these two strains, so he treated these strains into mice plural. So in case of mice, what he did is he first injected living R cells into mice. So after injecting this R cells, and again one more pointer, so R strain and the S strain is virulent, it is pathogenic, it causes pneumonia. Now it causes pneumonia because of its protective layer. So it is protection because of the mucus coat, so it is virulent in nature, it is pathogenic in nature and causes pneumonia. But R strain, it is not having any such kind of mucus coat, so it does not causes pneumonia. It is non-virulent, non-pathogenic, in other words. So what the Griffith experimented on this mice, he took living R cells, bacterial cells, and injected them into mice. So as R cells are non-virulent, non-pathogenic, so they do not cause any pneumonia, so the mice did not die. It is natural. Similarly, he took living S cells and injected into the mice. As S cells are virulent, they cause pneumonia, so naturally the S cells caused pneumonia in the mice, and thus the mice died of pneumonia. So it is also very simple. But in the third step, what he did is he took the S cells and subjected them to the steam. In other words, they were heat killed S strain, or by boiling the cells, so that the cells were made inactive. So these inactive S cells or heat killed S cells were injected into the mice, and as the S cells are inactive, they did not cause pneumonia, so the mice did not die. It is also natural, or easily we can predict. But in the fourth important step, what he did is he took the heat killed S cells which were inactive and mixed them in with the living R cells. So the living R cells and the heat killed S cells mixed and injected into the mice. But now what Griffith observed is the mice died of pneumonia. So though the S cells were heat killed, but they were mixed with the R cells and they died of pneumonia. So what by this what Griffith concluded is that there is some material or some substance, what he calls it as the transforming principle. So that is present in the heat killed S cells, and that transforming principle or that substance has been transferred to the living R cells, and thus the living R cells they became virulent because of this principle or the substance, and they caused pneumonia. That is what Griffith concluded. But Griffith did not explain about the nature of that principle, what actually that principle is, or what that substance or the material is which made the heat killed S cells transformed into R cells. That is, or the R cells, how they have been converted into virulent nature along with the heat killed S cells. So that is what Griffith did. But further what this Griffith is means he did not continue his experiment. Later after this, Avery, MacLeod and McCarthy, three other scientists, they continued this experiment, Griffith's experiment. It is a continuation of Griffith's experiment. So what they did is they started searching about the transforming principle what Griffith concluded. So in Griffith's experiment, what a transforming principle, so what is that a transforming principle, or what is that material or that substance which made the R cells virulent along with the, when mixed with the heat killed S cells. So what they did is they extracted the biochemicals from the heat killed S cells. So what heat killed S cells were present which are inactive, they extracted the biochemicals like DNA, RNA, carbohydrates, proteins, and these mixture of the biochemicals, they used different enzymes and treated on the mice, injected into mice. So first again what they did is the biochemicals were mixed with proteases and injected into mice. So when they were injected into mice, the mice died of pneumonia. That means the proteins were digested by proteases. So the proteins are not the cause of this or it is not the transforming principle. Again the biochemicals were mixed with the RNase enzyme and injected into mice. So again the mice died of pneumonia. So that means here also the RNA is not the cause or it is not the transforming principle. So in the next step what they did is they mixed the biochemicals with the amylases which digest the carbohydrates. So there also the mice died of pneumonia. That means the carbohydrates are also not the transforming principle, or it is not the substance. Then in the fourth step what they did is the biochemicals were mixed with the DNase enzyme, where the DNase which digests the DNA, and that when it was injected into mice, so the mice did not die. So that means when the DNA was digested, so there was no virulent nature, and thus the digestion with the DNase enzymes inhibits the transformation. So there was no transformation in case of this DNase, and thus this indicates that the DNA actually caused the transformation, not the carbohydrates, not the RNA, and not the proteins. So the DNA is the transforming principle, the DNA is the substance or the material which caused the transformation, and hence it was concluded that the DNA is the genetic material. So Griffith's experiment continued by Avery, MacLeod and McCarthy, it proves that DNA is the genetic material. So this part is asked in three types. Explain Griffith's experiment. Explain the experiment to show that DNA is the genetic material. Explain Avery, MacLeod and McCarthy's experiment. So Griffith's experiment is the same as that of Avery, MacLeod and McCarthy, which continued Griffith's experiment. So that is how they proved that the DNA is the genetic material. Thank you.